So our next speaker is Farhana Fathin, a student from the Gulf Medical University, UAE. So she'll be talking to us about a rare autoimmune disease, which is abbreviated as APECED, APECED. So over to you, Farhana.
Yeah, I'll share my screen right now.
Um, yes, your screen is.
Mm.
Yes, the poster is displayed in full screen.
So, good evening, everyone, and thank you all for joining me today. I'm Farhana Fathin Mahin Abubakar. I'm originally from India and I'm currently pursuing my undergrad degree in biomedical sciences from Gulf Medical University, United Arab Emirates. And I've been part of TBS for quite some time now, and I would really like to thank TBS for providing this wonderful platform. So, on that note, I'll be presenting a poster on APECED, or which is the full form of it, which is autoimmune polyendocrinopathy ectodermal dystrophy. So let's get started. So, I can either call it APECED or I can just, in short, call it autoimmune polyglandular syndrome type 1 or APS1. So they synonymously mean the same thing. And again, it's a rare autosomal recessive disorder, and it is caused by mutations on the AIR gene. The AIR gene is present at the locus of 21q22.3, and basically this AIR gene, it codes for an amino 554 amino acid protein. And the main function, there are some terms here, but when I start explaining what it actually does, I think it'll make all sense. So it is responsible for mediating a promiscuous gene expression in medullary thymic epithelial cells. So these thymic epithelial cells are present in the thymus gland. And if you know what the thymus gland is, it's a primary lymphoid organ found in the superior mediastinum above the heart, and it plays a very vital role in the maturation of T lymphocytes. And the full form of AIRE would be autoimmune regulator gene. So that is abbreviated as AIR, right? And what it does is this drives, and it drives a very essential process in building immune self-tolerance. So what is immune self-tolerance in these T cells that mature in the thymus gland? So immune self-tolerance is when a person's immune system, it does not respond to constituents of his or her body. So in the sense, your lymphocytes are supposed to know, because we know that lymphocytes are immune cells and they attack anything foreign, right? So our T cells should know what cells to attack and what cells not to attack. When uh, when there is a circulating population of lymphocyte in an individual that is not able to differentiate between the foreign and the self cells, so that is how you get autoimmune disorder. Like your body does not recognize its own cells and it ends up thinking it's foreign, and then you end up with an autoimmune disorder. So this is basically a very vague idea of what immune self tolerance is. And the defect on this gene, though it is associated with a system wide autoimmunity, but it is particularly directed at endocrine organs like the thyroid gland and the adrenal gland. And that is how the name suggests that it's a polyendocrinopathy disease. And it results in a classical triad of manifestations. And through studies, we have also come to know that the diagnostic triad has to be expanded as well. So, in the course of this presentation, these are the four main objectives that I'll be covering, which would be describing the mutations on the AIR gene leading to APECED, recognizing the classical manifestations of APECED, and revealing developments in defining more comprehensive diagnostic criteria, and also discussing some ongoing research in managing APECED. So first I'll discuss the causes and then I'll come to the symptoms, right? So it makes more sense. So now before we understand what causes this, we should know what this AIR gene actually does, or what is the function of this AIR gene. So we know that I just said that the lymphocytes, the T lymphocytes specifically, they mature in the thymus gland. So what is this process of maturation? What happens behind this maturation is that these lymphocytes that go to the thymus gland, they all undergo a process called negative selection in the thymus gland. So it's basically, you can say, a sort of course that these T lymphocytes go through, so then they can be released into all the other organs and they can perform their functions, right? So in this process, basically the T lymphocytes, they are taught by these thymus cells to how to recognize the self cells and not attack them. So they are able to differentiate between the self cells and the non-self cells. So how does this happen? You can say that the cells, for example, now if the T lymphocyte is being exposed to a thymic cell because it's in the thymus, so it will know a thymus cell. But what if this T lymphocyte has to travel to some other organ in the body? How will it know that this is my own heart cell and this is my own lung cell, or this is my own kidney cell, and that I'm not supposed to attack it? So this is where the fun part comes in. These thymus cells, they sort of dress up, like when we were kids, we played dress up, right? We dress maybe, fancy dress competitions where we dress up as another person and we try to talk like them, right? So this is exactly what these thymic cells do. What they do is they present some random TSA basically means tissue specific antigens on their surface. So if you take the heart cells, and if you take the lung cells, the cells in these organs will have some specific antigens on their surface. So what the thymus does is it knows all the major antigens, or the prominent antigens on those particular organs, and it will also dress up in that way. So basically the lymphocytes will know that this is so it will, if it is the thymic cell, it will show itself as maybe a cell in the heart or a cell in the lungs, right? So the cells in the thymus, they present random. It's really random. You cannot tell that only these type of tissue specific antigens are presented on the thymus. Like it's very random on their surface, so that these lymphocytes can recognize the self cell in any organ that they may be presented to in the future. So this AIR gene, what it does is it codes for the AIR protein, obviously, and this protein does not just work alone. It works as a complex, and it binds to the tissue specific antigen genes. So this protein goes and binds to the gene that is going to transcribe for the tissue specific antigen and initiate transcription. Initiate the transcription of the random TSA gene and produce these tissue specific antigens on their surface. And so then the thymic cells can undergo negative selections. So these the cells that express the AIR gene in them, so there are some cells that will express the so if I'm so sorry so any cell that expresses the AIR gene will express tissue specific antigen on them. And if the cell is not able to express the AIR gene, which is to transcribe for the AIR protein, then it cannot help in expressing the tissue specific antigen as well, because they are completely related to each other. So coming to the causes again, because it's a genetic disease, it's caused because of mutations. So as you can see here, this image shows the complete AIR gene. So this is your whole AIR gene, and it has 4 main domains. So I'll just go in this order. So first I'll discuss the domain and what are the mutations presented in that domain, and so forth, right? So the first domain that you have is So basically, these different domains, we know that when a protein is being transcribed, there are like so many processes that happen even after transcription, right? So all these domains play vital functions in the oligomerization of the air polypeptide as a whole, even in activation and gene transcription, etc. So first you have the caspase recruitment domain or the card domain. So the mutations, as you can see here, the mutations here would be PY8C and the PR139X. So I'll just give you a brief explanation of how these mutations are named, right? So P here represents, so this is like a general way how mutations are presented. So if you read this sort of code, maybe when you're reading some other article, you know what they're actually talking about, right? So the P here refers that the effect of this mutation is on a protein level. And here, and we know that uh, the transcription happens and then it each codon codes for an amino acid, right? So here Y is tyrosine, and we know that every amino acid has a one letter code. So Y here is tyrosine and C here is cysteine, and the number 85 here represents that the tyrosine in in the 85th position is replaced by cysteine. So this is how you understand how the mutation works, right? So this happens in the card domain, and it's mostly seen in Persian Jews. So this mutation can be mostly seen in Persian Jews, which is 1:9,000 is the ratio. And the next, another mutation that happens in the card domain itself is PR139X. So how do we interpret this? P again here refers to that it is the effect will be on a protein level, and R here is arginine, the amino acid arginine, at the 139th position, which is 139. X here represents the stop codon. So what happens here is the code that is supposed to produce arginine is being replaced with a code that is going to code for a stop codon. So in this case, what will happen? Your transcription will literally stop there. So you will, so if, only if the entire air gene is coded, you're going to get a complete air protein. So now, because it's going to stop in between, because there is a stop codon in between the gene, you will end up with there will be a complete absence of maybe the air protein, and more severe phenotypes of APECED is related to this particular mutation, and it is seen in sardin. I'm so sorry, Sardinians. Okay, yeah. And then that is about the card domain. And then you have the sand domain. The sand domain also has mutations in them. And again, if I think if you guys now, because I've explained the previous one, I think you should be able to understand how this one works. So here again, arginine is replaced by a stop codon in the 257th position, right? So because this is more downstream in the sense it's more towards the 3' end. So you can see that most of the protein will be, most of the gene will get transcribed, but at the end of the day you will end up with a more defective or a truncated non-functional air protein, right? And these two, especially PR139X and PR257X, they are important for initiating transcription, and therefore these two mutations are very crucial. So, and more more they're really associated with more severe phenotypes of APS. Then you have the plant PhD1 and PhD2. You have it towards the end. This is PhD1. Yeah, this is PhD1 and this is PhD2. And so here now you can see that this one looks quite different from the other three mutations we have seen before. Here, it first you can see that it says FSX. FSX here basically means that it is a frame shift mutation. So now, how do we read this? So the frame shift mutation is resulting from a 13 base pair deletion starting at leucine. That is what it says L here. At the 200 at the 323rd position, which is replaced by serine. right? And the frame shift mutation lasts 51 amino acid long. So that is why you have the number 51 here. And it's mostly this type of mutation is seen in North Americans, British, Norwegian, and Irish populations. So as I've mentioned before, that it is an autosomal recessive disorder. I'm so sorry. There has been cases. I think there was one case that was identified in Norway in 2001, and when they saw it, it showed that it was being autosomally, it was autosomal dominantly being inherited in the family, and that was not because of these four mutations, and it was completely due to a different mutation, which is G228. W, where glycine is replaced with tryptophan in the 228th position on exon 6. So majorly talking, it is an autosomal recessive disorder, but this particular mutation is it is inherited in a autosomal dominant pattern. So coming to the symptoms of this disease. So the symptoms of this disease have been majorly classified as the classical triad and the adjunct triad. So the adjunct triad was something that came into place after a lot of research and just recently, whereas the classical triad has been there for a long time. So, as again the name suggested, you have candidiasis, hyperparathyroidism, and adrenal insufficiency. Candidiasis Candidiasis is Candidiasis refers to an endogenous infection by the fungus called Candida albicans, and it is one of the major components of APS, and it appears in mostly about 97% of the patients by the time they each by the time they reach 30 years of age. And it's mainly manifested in the mouth as an oral thrush, and even in other places like the skin, the female genitalia, abdomen, and the intestine. Then you have hyperparathyroidism. Hyperparathyroidism, again, because it is an autoendocrine disease, it will obviously affect the parathyroid gland, and it is the first endocrine component, and it's developed usually after puberty. And the symptoms may remain vague for a long time with just slight muscle cramps, etc. So the immunological target in has been identified now as NALP5. So the NALP5 protein is the one that is targeted in the parathyroid gland, and that is what. So here you can see the autoimmune component over here, right? And then you have adrenal insufficiency, which is synonymously also called Addison's disease, and it's manifested as fatigue. You will have salt cravings, hypotension, etc. Then coming to the adjunct triad. So later in the presentation, I'll explain why was this adjunct triad introduced in this particular disorder, in this particular disease, right? So in the adjunct triad, you have urticarial eruption, which is you can say it's like rashes, which is seen as raised erythematous on the surface, and it will be seen. You can see this in uh, from like 3 to 36 months of birth. So you can detect it that early, right? And then you have intestinal dysfunction. Around 18 to 22% of patients with APECED uh, have some or the other sort of intestinal dysfunction. And that is because of the autoantibodies that are produced against tryptophan hydroxylase. So that is the reason why they get abdominal pain, cramping, bloating, distension, and also in some cases lead to vitamin B12 deficiency. Then you have enamel dysplasia. Dysplasia is basically when the teeth becomes yellow, white, and discolored, and you can see that the teeth is not, developing in a normal way. It will be pitted, misshaped, and etc. So you have to keep enamel dysplasia in mind because it plays a very major role in early diagnosis. And some of the other symptoms that might occur in these patients would be autoimmune pneumonitis, hepatitis, gastritis, and fat malabsorption. Then coming to the diagnosis and the treatment. So the diagnosis is basically, so when do you confirm that this patient has APECED or not? So if the person shows two of the three clinical manifestations of the classical triad, so they can have any of the two from the classical triad, then you would give them a diagnosis of APECED. So how is this characterized? So if you have hyperparathyroidism, obviously there will be decreased levels of calcium, and also the uh, when you run an assay of antibodies, you can find that if the presence of NALP5 autoantibodies will also indicate hyperparathyroidism in these patients. Then you have adrenal insufficiency, where the patients will have low cortisol and aldosterone with elevated ACTH and plasma renin. Then you have CMC, which is also which is the basically the Candidiasis infection. So you will also be seeing autoantibodies against Th17 cytokines, which will cause a decrease in IL-17 and IL-22. IL-17 is basically a pro-inflammatory cytokine that is needed for defense against bacteria and fungi. So because the levels of IL-17 is decreased, these patients become more susceptible to an infection by the Candida albicans fungi, which is an endogenous fungi that is usually present in our bodies. And now coming to why was this adjunct triad introduced in the first place? Like why was a need for an adjunct triad? So when they were analyzing the manifestations appearing in these patients for whom the symptoms of the diagnostic criteria did not develop as part of their first two manifestations. So the thing is, when there were a lot of patients who didn't show any symptoms from the classical triad, but they showed other symptoms. So this intrigued the researchers to find out more, evident symptoms that can, help them accelerate the diagnosis process. So as you can see here, that a study was conducted by Ferre and Rose et al. In 2016, which is quite recent, and it revealed that when integrating the classical triad and the adjunct triad, it was seen that the diagnosis was accelerated by approximately 4 years. 4 years is like a lot of time. So you could diagnose APECED 4 years early than what you usually the time it takes for it to be diagnosed, and this prevents. So this permits diagnosis before the development of much more severe conditions like adenosine crisis and hypocalcemic seizures, which may be fatal. And coming to the management, you have a lot of drugs that some of them are under clinical trials now. So these are the general drugs that are used. You have oral calcium and vitamin D supplements to mitigate the effects of low PTH, because obviously their parathyroid gland is being attacked here. So to mitigate the effects for that, and you have hydrocortisone and fludrocortisone. So again, because this is for treating the adrenal insufficiency, which will replace cortisol and aldosterone that are deficient in patients with adrenal insufficiency or Addison's disease. And you have azole and echinocandin drugs. These are for the treatment of CMC and omalizumab, which is a monoclonal anti-IgE administered to patients with urticaria for treating urticarial eruption. And also this CPP and CPP-ACP drug is also given to patients to treat them for APECED. So what is the current research that is going on in the field of APECED is mainly focusing on, there are other things, but I've decided to put these two, which is APECED pneumonitis and the treatment for CMC or the Candida infection. So if you see APECED pneumonitis, it was present in 40% of APECED cases. And most of the time it was misdiagnosed for something else. They just thought it was a normal pneumonitis, and it was not considered that it was caused due to the autoantibodies. Right. So what happened was, because it was misdiagnosed, this lead to the elevation of pneumonitis and leading to even more severe symptoms like bronchopulmonary fistulas, empyemas. And here you can see on the side, I've attached an image. So this is an X-ray image of a 9-year-old who came with a diagnosis of bronchitis along with an autoimmune lung disease. So here you can see she was administered with rituximab, and you can see the drastic difference in 6 months. This is in the February of 2015, and this is the August of 2015. So you can see a very drastic improvement in the lung condition. So this is one thing that's going about on research about more about APECED pneumonitis. And the last one is the CMC treatment, like how do we treat the Candida infection. So most of the patients, they started showing antifungal drug resistance, was also observed in these APECED patients. So because of this, they had to render the administration of azole drugs was considered to be ineffective, because these patients were getting resistant toward this drug, and it was not helping in controlling the infection. But in other cases now, quite recently, as you can see, as of 2018, there is this drug called VT1598. It's been proposed as a better alternative as for the azole drug, and also it also keeps in mind the IL-17 deficiency as well. And also, it is not associated with any renal toxicity, unlike azole drugs. So, azole drugs was also shown. Azole drug was also caused renal toxicity in patients. And the good part is that the recent studies on BT1598 are being conducted on mice, and successfully the drug has entered phase 1 human clinical trials as of 2018. So this is all about APECED. And just to summarize, so they are also the main research going on behind this is to keep expanding the diagnostic criteria, so it allows early diagnosis. And more research is still needed on the mechanisms of development of APECED symptoms in order to improve management protocols. And some ongoing investigations being conducted by the NIH include pathogenesis of APECED related GI diseases, because we said that the adjunct triad includes intestinal dysfunction, and also about prophylactic immunomodulatory based therapies, and also studying the causes of CMC susceptibility in APECED patients. So this brings me to the end of my presentation. And once again, I would like to thank all the organizers and TBS team for this opportunity, and my other fellow speakers as well for sharing your valuable knowledge, and to all of you fellow listeners here for giving me your time, because it really means a lot. And also on this note, I would like to thank my friend and my fellow batchmate, Maha Hassan, who also contributed majorly to the content and the design of this poster, and without whom this would have not been possible. Thank you so much, Maha. And once again, thank you all, and have a great evening ahead.
Thank you very much, Farhana. That was a really good
presentation.
You broke down the concepts and delivered it to us. It was really effective. Yeah. So, the audience, you can put in your questions through the chat, or feel free to unmute and ask. I wanted to ask about does this disease affect the chances of survival? Like, how fatal is it?
Sorry, I couldn't. Could you please repeat yourself?
Is this life-threatening or just like debilitating?
I think it depends on the severity, and again, because it's a genetic disease, it can only be managed, right, to a certain extent. So I think it would put a pressure on your day-to-day lifestyle. And yes, it depends on the severity. So, but like as some particular components of it might speed up really quickly and worsen the disease to an extent that you cannot even manage it, sort of.
So what made you come across this disease in the first place?
So basically, this poster that we did was, like I said, me and my friend. What we did was, it was presented as part of our course. We had to present it as part of our human genetics course as a seminar. And we had to take any genetic disorder. So she was the one who came up with this, and even autoimmune disorders. I don't know. I find them really interesting to learn more about them. So yeah, so that's the reason why we took this, and it was pretty rare. And like there were many other diseases, but like this one was not too heard of, and it's under research right now. So yeah.
Interesting. Okay. If there are no further questions, then we'll move ahead. Thank you once again, Farhana. It's really nice. And the poster
Thank you
is
so much.
really nice. The
Oh,
template.
thank you.